So as you said, I'm the head of the Department of Pharmacology at the University of Auckland, but I'm also a cannabinoid researcher. I did my PhD a long time ago when the first cannabinoid receptor was discovered. And so I've had 20 years of cannabinoid research since then. And what I was told to do today was to give you the Pharmacology 101 on cannabis and how it works. And I know it's a very diverse audience, so some of you will know all of this already, and some of you will think, why on earth is she showing us cells? But bear with me, because what I wanted to do was just talk to you about how drugs work in the brain, and then how cannabis works within that context. Because if you understand how drugs work in the brain, it's really easy to see how cannabis is just another one of these drugs. And I took too much advantage of the free coffee this morning, and I'm very over-caffeinated, so this is going to be waving around wildly. And I'm going to point on this side, so I hope you guys can see it. And I have to point this at them. All right, so how does the brain work? Sorry, that's reproducing really badly up there. Brain cells talk to each other. And what we have, what you should be able to see here is a cell body putting out an axon, and that axon is terminating on neighbouring cells. And that's how all brain cells communicate with each other. You have these points of contact between them. And if you look at the point where they connect with each other, which is magnified here, what you have is this terminal button. And so essentially, you've got the vesicles containing neurotransmitter, which spill out, and cells communicate across the synapse, which is the space between two cells. Pointed at them. And so this is a really highly stylized nerve terminal. This could be any neurotransmitter in any nerve terminal. And so it's got the components of all nerve terminals rather than being specific for one. And if I just take you through the real basics of this, because as I tell my undergraduate pharmacology students, if you can understand this, you can understand how almost any drug works in your brain. So essentially what we have is a precursor molecule which gets synthesized by an enzyme into a neurotransmitter. And so that could be dopamine, glutamate, GABA, any of the common neurotransmitters, or it could be an endocannabinoid. That then gets packaged into vesicles, which gets stored in the nerve terminal. And when an action potential comes down this nerve terminal, it activates channels, which lead to that neurotransmitter vesicle fusing with the cell membrane and spilling neurotransmitter out into the synapse. And when it's in the synapse, that's when you act on receptors. So there'll be receptors on the postsynaptic cell, and there's also presynaptic receptors, and the neurotransmitter will act either on these receptors to activate them or presynaptically to actually inhibit further release. And there's always an off signal for a neurotransmitter. They're there and they're gone very, very rapidly. And the off signal is one of two options. It's usually either an enzyme, which is in the synapse, which breaks it down really quickly, or it's a transporter, which takes the neurotransmitter back up into the presynaptic synapse so that it's gone away from the receptors. So most neurotransmitters are present for just milliseconds within the synapse. And sometimes that degradation product will also be a precursor that can be carried into the nerve terminal and used to make more. And so if we think about how drugs work, there's four main sites of drug action: ion channels, enzymes, transporters, and receptors. And almost all drugs act on one of these protein targets. The only real exception are the odd, some drugs which act on DNA, but most drugs will act on one of these four protein targets. And so if you just look at those in turn, if you were to act on the ion channels to inhibit them, then what happens is an action potential comes down the synapse. You no longer get the, if the ion channel's blocked, you don't get this vesicular fusion, and then neurotransmission won't occur. The neurotransmitter won't be released. If it's acting on an enzyme, such as, for example, the degradation enzyme, then if you inhibit this with a drug, you're going to have neurotransmitter present in the synapse for much longer. You're going to have much more effect on your receptors. You might even get it diffusing onto neighbouring cells. And so in the case of acetylcholine, your acetylcholine esterases would act on this enzyme, and you would get far more acetylcholine, and that's how most of the nerve gases work. In terms of transporters, this taking the neurotransmitter out of the synapse. If you inhibit that, you also end up with a lot more neurotransmitter present in the synapse. And you'll probably all be familiar with drugs that do that, such as the selective serotonin reuptake inhibitors, SSRIs, your antidepressants. So they're simply acting by inhibiting this, and then you've got more serotonin in the synapse. And finally, you've got receptors. So a drug can act to bind to the receptors and activate them, or it can act to bind to the receptors and prevent their activation by your natural neurotransmitter. And so an example of a drug that would act through a receptor that you'd all be familiar with would be something like morphine, which acts at the receptor for the endogenous opiates, at the mu-opioid receptor, to mimic its effect. And so if we look at the history of cannabinoid research, really, the modern history over the last 40-odd years, it really all began to gain a real scientific basis in 1964 when Rafi Meshulam's lab at the Hebrew University isolated and elucidated the structure of delta-9 THC. And if you read the early reports of it, he says him and his friends took it and agreed that this was just like taking cannabis and that this was most likely the main psychoactive ingredient in cannabis. And then it was a long time before anything much more happened. And the reason for that is in the lab environment, delta-9 THC is horrible to work with. It's really lipophilic. It's not at all water soluble. It sticks to plastic and glass in your test tube. So it's very, very challenging to work with. And so it really was quite a long time before any really significant progress was made in the field of how THC actually works. There was a group in 1981 that showed that it had stereoselectivity. And that's a classic sign of something that acts at a receptor. Most drugs that act on receptors have stereoselectivity. So that was really the first suggestion that maybe it was acting specifically at a receptor. And then we had the synthesis by Pfizer of CP55940, one of the synthetic cannabinoids, that has slightly better properties in terms of handling it in the lab, slightly more soluble than THC. And that actually led to quite a few more studies being done trying to understand the mechanism of action. And using this compound as well as a couple of others, including THC, Bill Devane in Lynn Howlett's lab was able to show that there was signal transduction occurring that was very consistent with the action, again, at a receptor. So it was strongly suggesting that there was a receptor. And when we continue, the really big breakthrough in cannabinoid research came in 1990. And it really was almost random chance. It's quite a nice story. There were two groups working at the Lab of Cell Biology at the National Institute of Mental Health, working so within the same department. Miles Herkenham, who'd got hold of a radioactive version of this Pfizer compound and was trying to map the binding site for cannabis in the brain. And this is one of his images, where you can see that there's just bucket loads of binding site. The brighter it is, the more receptor there is. His suggestion was it's actually the most highly expressed receptor for a neurotransmitter that you have in your brain. And at the same time, Lisa Matsuda was trying to clone a substance P receptor. And so she was using homology cloning strategies, just looking for something that was like a substance P receptor, but not a substance P receptor. She pulled out something that looked like a receptor, and much to her great disappointment, it didn't respond to substance P or substance K or any of those substances at all. So she hadn't found at all what she was looking for, but she mapped her receptor in the brain, and luckily, having Miles a floor below, they sort of put the two together and thought this looks like it might be the same thing, and tested out her receptor and discovered that what she had in fact found was a receptor to cannabis. And so really, had this not been happening in the same building as each other, I suspect that the cannabinoid receptor could still be an orphan receptor and we could still be looking for it. And so that she had turned out to have found a cannabinoid receptor, which we now call CB1. So in general terms, So far in biology, we don't have receptors in case we randomly wander across a field and think I might smoke this plant. We generally have receptors because we have neurotransmitters that recognize them. And so just as would be predicted, we have endogenous cannabinoids. And interestingly enough, it was back to Rafi Meshulam's lab at the Hebrew University who found the first endogenous cannabinoid, anandamide, which they actually found in pigs' brains. They'd figured out it was likely to be something quite lipid soluble, so they'd taken quite a different approach to what had been used before. And this is it here. It's actually arachidonic acid with an ethanolamide attached to it. So it's a really unusual structure for a neurotransmitter, which also probably accounts for why it hadn't been found earlier. A second cannabinoid receptor was then cloned, and this was found to be really highly expressed on immune cells. A lot of debate as to whether it is actually in the brain or not in the brain. I'm on the camp of it not being in the brain, so I'm not going to talk much more about it because I don't think it's mediating any of the psychoactive effects of cannabis. And a second endocannabinoid was then found as well, which is very similar. Again, it's arachidonic acid, but this time it's got a glycerol attached to it. So we now have 2 endogenous cannabinoids, we have 2 cannabinoid receptors, and so hopefully you can start to see how THC might be acting. And that is that the CB1 receptor and its CB2, in fact, are G-protein-coupled receptors. And I don't want to go into huge amounts of signaling because your eyes will all glaze over and half of you will fall asleep because I think there was a dinner last night as well. But CB1 being a G-protein-coupled receptor is exciting from a pharmacological point of view because G-protein-coupled receptors are very druggable. And so this is medicines with FDA approval broken down into category as to what they actually target. And so you can see that they started out with 21,000 drugged products that currently had FDA approval. This was in 2006, but it hasn't really changed much since then. They broke that down to what were actually unique products, so took the salts of different things to be the same thing, and then looked at what they target. And when you take out the targets that aren't actually in the person, so lots of drugs are targeting the bacteria or the virus, what it ends up with you having is 266 human targets, which I thought was remarkably small. But if you look at this, well over a quarter of them, and I'm really sorry, I don't know why none of my slides are reproducing. They look fine on this screen here. Well over a quarter of them are actually G-protein-coupled receptors. So this is a really interesting target from a pharmaceutical perspective. We know that we can design drugs which target G-protein-coupled receptors because we do. There's well over a quarter of the market is actually targeting GPCRs. So, a little bit more on how drugs work at receptors. THC is an agonist at CB1. And what do I mean by an agonist? It means that it binds to the receptor and it activates it. So in other words, it mimics the endogenous neurotransmitter. All of your endogenous neurotransmitters are agonists. And when we measure biological response, we measure 2 components of it. The first one is efficacy and the second is potency. And so efficacy is the ability of the drug to produce an effect, how big an effect it produces. So the maximum effect would be a drug which is classed as having high efficacy, whereas potency is defined as the concentration of the drug that it takes to produce an effect. So we define that as the concentration that it takes to produce 50%. And so we compare the 50% concentrations across drugs to see which is more potent. So you can see that if it takes less drug to produce an effect, then it would be considered to be more potent. And that should be illustrated on this slide, with the one drug is almost disappearing here, where this drug would be considered to be more potent because it takes less concentration of it to produce its response, whereas this drug is more efficacious because it reaches a higher response. So less potent, but more efficacious. And so where does THC fit into this with the cannabinoid receptor? To look at the biological responses, it's worth just looking a little bit at what sort of a receptor this is, and I'm not going to go into all the in-depth signaling pathways here. But in general, the drugs, the agonists binding to the receptor, activate a range of different pathways, and we can measure different steps of this. So they activate G proteins. CB1 is actually a GI-GO link, so it activates a G protein that inhibits adenylate cyclase and cyclic AMP formation and activates inwardly rectifying potassium channels. And in response to agonist binding, the receptor gets internalized and actually degraded. And so what I wanted to show you is just some work from our lab and from other labs looking at THC and a range of other agonists acting at the receptor. And so what I've got next. No, sorry, that's awful. Is a series of synthetic agonists. And what I'd hoped you would get to see here is the structures of them. I don't know if you can see that at all. Essentially, within the synthetics, there's a couple of different key structures. Those that are like THC, which the example I would be giving you, if you could see it here, is CP55940. And those that are aminoalkyl indoles, which are like WIN55212. And that's been the backbone for many of the synthetic compounds on the market currently. We have then partial agonists, which you can't see, but which I've included THC in, and I'll show you why, and the endocannabinoids. And so if you look at this range of different compounds' ability to actually activate the receptor, what you can see here, or you could see, is that THC is actually a weak partial agonist. And what we mean by that is it's very low potency and it's not very efficacious. So what you would be able to see if this graph actually had lines on it is that these lines here show CP55 and WIN55212, which are the synthetic cannabinoids, and they're very efficacious and they're very potent. This line down here is actually THC. This is an activation of the G protein, but this is THC here. And so what you can see is it takes a lot more of the THC to activate the receptor, and then it activates it very inefficiently. So it's activating only 20% of the maximum possible effect. And that's even at very high concentrations. So this is up at 100 millimolar here. And likewise, at least this one you can see, this is just another example. This is the ability of them to activate inwardly rectifying potassium channels. And again, this is CP55940, one of the synthetics. In red is the WIN compound, another synthetic cannabinoid, and in green there is delta-9 THC. And so you can see that again in this, it's very low potency. It's almost 100-fold less than the synthetic cannabinoids, and it's very low efficacy. So it really only activates a portion of the total possible response through the receptor. And I know we've got a session on synthetic cannabinoids coming later, and I suspect that this might account for some of the differences that people experience with them. But just to finish off with one more unreadable slide, what I wanted to sort of leave you with is the idea that from a pharmacology perspective, we're interested in the cannabinoid system well beyond the receptors. Just like other neurotransmitters, there are a wealth now of different possible drug sites that have been thrown up by the discovery of the endocannabinoid system. So what we have here is the receptors, obviously, also the endogenous cannabinoids, 2-AG and anandamide. And with them, there'll be a whole range of different drug targets. If you think back to where we started, that drug targets are receptors and ion channels and transporters and enzymes. There's enzymes that are involved in the synthesis and the degradation of the endocannabinoids. There's transporters involved in releasing them into the synapse. And then obviously there are also the receptors, which have been of interest as well as potential drug targets. But there is quite a lot of work going into the potential of actually altering endocannabinoid levels from a pharmaceutical perspective, which are approaches that can work with other compounds. So from a purely pharmacological viewpoint, the cannabinoid system is far more than just CB1 and potential psychoactivity. There's a lot of scope there for targeting other components as well. And with that, I'll thank you for your attention.
See, and you thought Friday morning was just going to be cruisy, kind of not using your brain too much. There you go. Have we got some questions, please? Over there, sir. Get a microphone to you. There you go.
Thanks for that, Michelle. That was a really great presentation. Having sat in on a few pharmacology things, I felt a bit primed, and so I feel even more aware of it now. Have you, or are you aware of any work that's been done with cannabidiol, with CBD?
Yep, quite a lot. So cannabidiol is a really poor CB1 or CB2 agonist. It's it will activate them eventually, but it's even less potent than THC. There's quite a lot now coming out suggesting it might be targeting serotonin receptors. I haven't seen any really good convincing basic pharmacology, but there was just a paper over the last couple of weeks that was suggesting that some of the potential action on cancer cells was coming from targeting one of the serotonin receptors. Can't remember which subtype. But that is looking like the most likely target at the moment.
Any questions? Thank you. Just down the front there, please.
Michelle, I'm just wondering why it is that you're of the school of thought that there are no CB2 receptors in the brain?
Well, because we can't find them. So initially, when the CB2 receptor was cloned, they did Northern blot analysis and everything else and suggested that it wasn't in the brain. And since then, there's been predominantly one group that couldn't find the mRNA but did find the protein, which would be a wonder of science if there is in fact protein without there being mRNA there first. But they've managed to publish a lot on that, and it really has strongly suggested, and we've published in this area, that the antibodies are picking up something quite different. And there's a lot of papers out now showing that the antibodies are picking up exactly the same pattern of staining in knockout mice who don't have the CB2 receptor, but their brains are lighting up with their CB2 antibodies. So we've done quite a bit of work, because it is on immune cells, and so the suggestion is that it possibly is on immune cells in the brain, so microglia, astrocytes. I believe it may be in rodent brain. In the human brain, we can't see it right now on the immune cells. We do see it on blood vessels. So when I say it's not in the brain, I mean it's not on brain cells. We do see it in blood vessels in the brain. We may be wrong still, but if it is there, it's at very low levels. Because you can do PCR, and you have to do repeated cycles of PCR to pick it up, which would be like contaminating your brain with one blood cell, basically.
Hi. Does the fact that the synthetics are full agonists mean that they are necessarily not as safe as a partial agonist? And does that have any implications for the new psychoactive substances bill?
It certainly means they're going to have quite different effects. One of the interesting things, and this may be going too far into pharmacology, is that because THC is such a weak partial agonist, it actually will, if you have endogenous cannabinoid present, it would antagonize it, because it binds to the receptor and has less effect per binding event, if you like. So some of the effects that you see might actually be antagonism of the receptor rather than activation, whereas obviously with a full agonist, that's not going to be the case. You're going to get often an even bigger effect than you would get for your endocannabinoid. So it's certainly likely to account for some of the differences. Quite whether that translates into safety is a different issue. But what we do see, for example, is in response to the synthetics, we get really massive internalization of the receptors and degradation of them. Whereas with THC, you actually don't get that much internalization or desensitization of the receptor. So, you could get quite different downstream effects of it.
Just a question. There we go, back there.
My understanding is there's more than just delta-9 THC, and I just wonder whether other cannabinoids have modulating effects, or...
Yep.
I mean, the thought at the moment is cannabidiol has the modulating effect, but I just wonder whether there are other cannabinoids that might also...
So I guess the current thinking right now is more along the lines of the other cannabinoids are probably changing your pharmacokinetics, and I haven't talked about the pharmacokinetic side of it. So they're probably changing how much of the THC actually gets to the receptor, because they'll be changing they're being metabolized by similar enzymes, and so changing the metabolism. Whereas most of the other cannabinoids don't appear to be having effects on the actual cannabinoid receptors. And those that are suggested to have activity, such as CBD, it looks like they're going to have their own receptors that they're targeting. So they're going to be doing the same sort of thing, but probably not at a cannabinoid receptor.
Question over there.
Is that going? Yes. Just wanted to ask, you said the receptors get degraded through the THC. So that's damage that happens at the cellular level, is that right?
So we don't consider it damage, it's normal regulation of the receptor, but it probably accounts for some effects on tolerance. So one thing we would expect is that you might develop tolerance more rapidly to the synthetic cannabinoids, because you get really rapid internalization and degradation just of the receptor, not of the cell. So in general with receptors, almost always in response to agonist stimulation, they're desensitized, so they stop responding and then they internalize. And then some receptors recycle back to the cell surface quite quickly and you become responsive again, and other receptors degrade and you have to make more receptor before you become responsive again. And with the cannabinoid receptor, at least in our lab and in a few others, it degrades quite rapidly and you actually have to make more receptor to become responsive again. So it's just a mechanism by which tolerance is probably being mediated.
Any last questions?
That would mean that the synthetic cannabinoids are much more addictive.
Certainly that you would develop tolerance more rapidly, and that is often linked, I think, with the addictive potential.
That's great. Thank you very much.
Thank you.
Michelle Glass.